Hello, everyone, and welcome to ACS webinars, connecting you with the best and brightest minds in chemistry, live from Washington, D.C. I'm Russell Silber, and I am pleased to be your host for today's broadcast, which is being co-produced with the ACS Division of Chemical Toxicology, the ACS Division of Medicinal Chemistry, the American Association of Pharmaceutical Scientists, and ACS Publications. Today, we will continue our series on the mitigation of drug-induced liver injury with a team of chemists from Merck. Michael Hafey, who is a principal scientist, Wen Kang, a director of assay development, and James Monroe, a senior principal scientist. We would love to get you involved in the discussion. You can share your thoughts and questions in the questions panel, and you can also join the discussion on Twitter by including @AmerChem Society in your tweet. The slides for today's webinar can be accessed right now by clicking on the link that you see in the GoToWebinar control panel, and also in the GoToWebinar control panel. An invitation email to view the unedited recording from today's presentation will be sent to all registrants for a 24-hour period, after which it will be removed, edited, and become an ACS member benefit. Our moderator for today is Kaushik Mitra, who is currently director in the Department of Drug Metabolism and Pharmacokinetics at Janssen Research and Development, based out of Springhouse, Pennsylvania. In addition, he serves as the head of the Biotransformation Group and the DMPK lead of the Cardiovascular and Metabolic Disease Therapeutic Area, overseeing an active portfolio of multiple internal and external assets. But before I turn things over to Kaushik, we'd like to learn a little bit about everyone tuning in today with a poll question. And you can select the choice that best describes you using the radio buttons on screen. So the question is, what is your current experience with drug-induced liver injury? And you can select one of the following. Are you a medicinal chemist and encounter DILI in your career, in your research? Are you a clinician and observe DILI in your patients? Are you a patient yourself and have been personally affected by DILI or other? And you can let us know more about that in the questions panel. And as we wait for everyone to choose their option, I'd like to remind everyone that during the presentations today, you can use the questions panel to submit questions for Michael, Wen, and Jim. And we ask that you submit your questions early and often to have a good chance to have them asked in the live Q&A after the presentations are over. So as people turn in their responses, I'm going to wrap things up here in 5, 4, 3, 2, 1. So it looks like the majority of responders are medicinal chemists who are working or encountering DILI in your career. And then we have a few people who are clinicians and patients themselves. And then the other vast majority, it looks like, is in the other. And we will look for those responses in the questions panel to share with our presenters. So thank you all for joining us today. We are excited to hop into our webinar, and I will now hand things over to Kaushik to get us started for today.
Excellent. Thank you so much for getting us started here. First of all, I'd like to welcome all our audience from across the world and the speakers for today, Mike, Wen, and Jim. I've had the privilege of working with all of them on a personal basis for many years, and I can vouch for their the scientific excellence in the field of metabolism and toxicology. So I feel really blessed to be able to coordinate and monitor this program with them. To start off our discussions today is Mike Hafey, who is a principal scientist in the Transporters and In Vitro Technologies group within the DMPK department at Merck. Mike received his BSc and MS degrees in biology from Villanova University and joined Merck in 2004 as a staff biochemist. Over the past 16 years, he has been a very strong contributor to establishing novel in vitro assays to measure transport by various intestinal, renal, and hepatic drug transporters. He has also developed and implemented higher throughput automated centralized PGP, permeability, and BSEP inhibition screening assay. Currently, he is regarded as the transporter subject matter expert on drug discovery and development program teams within Merck and externally as well. And his role is to elucidate what transporters do in drug disposition and assess the risk for transporter-mediated drug-drug interactions. He is also a member of the cross-functional team that explores potential linkage between BSEP inhibition and drug-induced liver injury, which is the topic that he will be speaking about today. And he has driven the development of a BSEP inhibition de-risking strategy utilized within Merck and has published papers on this topic as well. Over to you, Mike.
Kaushik, thank you for that kind introduction. Can you see my screen?
Yes, we are good to go.
Okay, awesome. So thank you, Kaushik, for the introduction. Thank you for the organizers for inviting me today to speak. As Kaushik mentioned, my name is Michael Hafey. I'm a member of the Transporters and In Vitro Technology Group within PPDM at Merck & Company. And I'd like to discuss with you today our two-tiered approach to de-risk drug candidates for potential BSEP inhibition liabilities in drug discovery. And the content of today's presentation was recently published in DMD. So I hope by the end of my webinar that you'll be able to identify key hepatic transporters involved in bile acid disposition, as well as understand the correlation between BSEP inhibition and DILI risk. Finally, I hope you'll learn how to utilize a two-tiered in vitro approach to limit compounds that may inhibit BSEP in vivo from reaching the clinic. This is a brief outline of my webinar. So I'll give you a high-level overview of hepatic bile salt transport, as well as introduce you to a decision tree that we're currently using within Merck to mitigate BSEP inhibition potential. I'll be talking about two in vitro assays, so a vesicular inhibition assay, as well as an hepatopac transporter inhibition assay that we use to de-risk BSEP inhibition potential. I'll be going over experimental background as well as study design, and then I'll give you some example data sets, and then wrap up with some conclusions. So drug-induced liver injury has been the most frequent single cause of safety-related drug marketing withdrawals for the past 50 years. DILI often emerges in late clinical development or post-marketing. The causes are poorly understood and are often multifactorial. As such, there's no single assay available to de-risk for DILI at the preclinical stage. Instead, we use a battery of assays with a weight of evidence approach. Disruption of hepatic transport proteins involved in bile salt transport is one potential mechanism that can contribute to DILI and will be the focus of my talk this afternoon. So the enterohepatic circulation of bile salts is complex. It involves multiple transporters at the level of the intestine and the liver. If we focus on the liver, bile salts are synthesized from cholesterol in the hepatocyte and subsequently effluxed across the canalicular membrane of the hepatocyte, mainly by BSEP, with MRP2 playing a smaller role. About 90% of bile acids effluxed into the bile are reabsorbed in the gut and make their way back into the plasma through the intestine. These recirculating bile salts can then be taken up mainly by NTCP, with OADP playing a smaller role, and returned back to the hepatocyte. Now, in regard to the driving force for generation of bile flow, it's really BSEP-mediated efflux that represents this driving force. And the mediated efflux is the rate-limiting step in overall bile salt transport. Interference with the efflux of bile salt via BSEP inhibition from hepatocytes could lead to increases in intracellular concentrations of bile salts, which can lead to toxicity. Now, the cell is equipped with compensatory mechanisms to help deal with this. Most of them are regulated by FXR. And so what you'll see is that when you have a rise in intracellular concentrations of bile salts, you will see a downregulation of the uptake transporters NTCP and OATP, such that you bring less bile salts into the hepatocyte. You see an upregulation of BSEP and MRP2 on this canalicular membrane, such that you pump bile salts into the bile out of the cell. And then finally, you see an upregulation of efflux transporters that sit on the cytosolial membrane of the hepatocyte, namely MRP3, MRP4, and OST alpha beta, and they will pump the excess bile salts from inside the hepatocyte out into the plasma. Now, even though the cell is equipped with these compensatory mechanisms, there can be conditions where the body fails to adapt, and BSEP inhibition and the increases in intracellular concentrations of bile salt in the hepatocyte lead to toxicity. And recent studies have shown that several drugs implicated in DILI inhibit BSEP. Because of these findings, we sought to understand the correlation between inhibition of liver transporters involved in bile salt transport and human DILI risk, as well as to establish in vitro assay systems to measure the effect of test compounds on bile salt transport. And we did this by taking two different approaches. So we studied inhibition of BSEP, MRP2, 3, and 4 in membrane vesicles using a test set of about 120 DILI positive and DILI negative compounds. We also developed and characterized a holistic taurocholate transport inhibition model in a long-term human hepatocyte micropattern co-culture model, which is known as HepatoPac. This work culminated in this BSEP inhibition decision tree that we've adopted within Merck. And I don't want you to focus on the intricacies of these boxes at this point. I hope that as we go through this presentation, you'll learn how we're making decisions to work our way down this decision tree. But rather, what I'd like to point out is that the decision tree is made up of two in vitro assays. So we have a BSEP vesicular inhibition assay towards the top. This assay is run at Merck in two modes. So we have a 3-point screening assay. We can run this in lead ID or lead op, give the chemist a chance to get a feel for whether or not they may potentially have a liability around BSEP inhibition. Also, they can explore some SAR. We also use this assay to generate full IC50s for BSEP, and all of our single candidates before entering into the clinic, we have a full IC50 in vesicles. Now, for compounds that have a perceived risk in this vesicular inhibition assay, we have a downstream HepatoPac TCA inhibition assay that we can then run. And we'll talk about the intricacies of that assay and how we can make decisions off of that in a few minutes. The other thing I'd like to point out is if you have a perceived risk in the vesicles as well as in HepatoPac, you would end up in this bottom right-hand corner. And this box, it says potential to inhibit BSEP in vivo, consider SAR to reduce the liability or clinical monitoring for cholestasis. The point I want to make here is that we're not killing compounds due to BSEP inhibition, right? We know that DILI is multifactorial, and we're really taking a weight of evidence approach. So while we're assessing BSEP, in our DILI de-risking, we are also looking at other things like mitochondrial toxicity, formation of reactive metabolites, and all of this gets put together to make a decision, on whether or not a compound would proceed to go forward. So BSEP inhibition does not necessarily kill a compound from our standpoint. So now I'd like to jump into the first in vitro assay, our vesicular inhibition assay. So in this assay, what we're doing is we're using inside-out membrane vesicles containing BSEP to explore the ATP-dependent uptake of a probe substrate, taurocolic acid, in the presence or absence of potential inhibitors. And we're basically asking the question, does compound X inhibit BSEP in vitro? It's very simplistic. We're taking the drug and putting it right, at the site of the transporter, and we just want to see if the drug interacts with BSEP and inhibits the uptake of taurocholate into this vesicle. This is an example of data from this assay. So throughout my talk today, I'll be showing data from MK866 to illustrate some of the points that I'm making. MK866 was a GPR40 agonist that was stopped in clinical development due to liver enzyme increases. We retrospectively went back and put this compound in all of our DILI de-risking assays. So of course, we obviously looked at BSEP inhibition. So in panel A, you're just looking at the positive control. And so what you can see on the y-axis is TCA uptake into the vesicle in the absence or the presence of atorvastatin. And atorvastatin is just a BSEP inhibitor. And so what you can see is a significant increase in the ATP-dependent uptake of TCA into vesicles containing BSEP in the absence of atorvastatin compared to that in the presence of atorvastatin. In panel B, what you're looking at is TCA transport on the y-axis again. But on the x-axis, you're looking at increasing concentrations of MK8666. And so what you can see is a concentration-dependent decrease in the ATP-mediated uptake of TCA into the vesicles. We can then normalize to percent control, where the uptake in the absence of the test compound is defined at 100%. And we can plot percent control versus MK866 concentration on the x-axis, and we see that this compound has an IC50 of around 0.8. So the next question should be, well, how do we put this 0.8 micromolar IC50 into context? And so what we decided to do is we ran 121 compounds that were either DILI positive or DILI negative in our BSEP inhibition assay and generated IC50s. So 64 of the 121 were DILI positive compounds, 57 were DILI negative compounds. And you're looking at those results in this graph on the left-hand side of my slide. So on the y-axis, what you're looking at is 1 over the estimated IC50. So the more potent you are, the higher up you are on this y-axis. And on the x-axis, you're looking at our exposure parameter. So now the exposure parameter that you'd really like to use to put BSEP inhibition data into context is unbound intracellular concentration. Now, this is a really difficult number to get at. So as a surrogate to that, what we used was calculated unbound concentration at the inlet of the liver. This really essentially equals your unbound Cmax plus the fraction of the dose absorbed. And this number was calculated for each one of these parameters using the highest clinical Cmax, the highest clinical dose, along with the associated Cmax for that dose. So we generated our IC50s and we put them on the graph according to the unbound inlet concentration. The green circles are DILI negative compounds and the red X's are DILI positive compounds. And what you can see is that you get an aggregate of DILI positive compounds in the upper right-hand quadrant of this graph. We then drew an empirical line to maximize specificity. So this is just an empirical cutoff that we're using. And the idea being that inhibition of BSEP may be predictive of human DILI for compounds that fall above this line up here. But inhibition of BSEP is not thought to be predictive of human DILI for compounds that fall below the line. I should also just point out that this line, so, if you think about trying to put data into perspective in, say, lead ID or lead op, you don't always necessarily have dose predictions at that stage. If you take a look at this line sits right at about 5 micromolar. So if you're unable to calculate your free inlet concentration, you can use 5 micromolar as a static cutoff. And the assay performance is about the same when you look at the sensitivity of it. So when you take a look at the sensitivity analysis, what you can see is that we have a high rate of specificity. So we only have one false positive. It's a compound up here called diprimidol. And we have low sensitivity, right? This is because you have a lot of DILI compounds that fall below the line. This is not really surprising. So, we already said that DILI is multifactorial. There's multiple mechanisms. So anytime you're looking at something that's multifactorial, but only looking at one endpoint, your sensitivity is likely going to be low, right? So the thinking here is that these red X's, the DILI could just be caused by a different mechanism, right? So that's how we put our data into context. And so if we go back to MK866, we know that the IC50 was around 0.8, so 1 over that IC50 would be 1.3. We had dose predictions, so we can figure out the unbound liver inlet, and you can see that this compound clearly falls above this empirical cutoff. And so this would be flagged for further investigation in downstream assays that we'll talk about in a second. So before going to our secondary in vitro assay, I did want to take a second to just talk about the role of MRP2, 3, and 4 inhibition. So we often get asked, if you remember at the beginning of the talk, I mentioned that MRP2 aids BSEP in the efflux of bile salts into the cannular space. MRP3 and MRP4 can be upregulated, and they will move compound from inside the hepatocyte across the cytosolic membrane into the plasma. So we get asked, okay, well, what if we combined BSEP plus MRP2, 3, and 4? Are we better at predicting DILI? And so what you're looking at here are assay results in our vesicular experiment for BSEP, MRP2, 3, and 4 vesicle inhibition experiments. The red is a positive, so this is where you'd be above that cutoff, and the green is a negative, so you'd be below the cutoff. And you're looking at those results from the DILI negative and the DILI positive compounds. And so what you can see from this is that a compound positive for BSEP, regardless of what's going on with MRPs, may be a DILI risk. And this is indicated by the fact that we only have one false positive. We only have one DILI negative that's above that threshold. When you start to look at MRPs alone or MRPs in combination with BSEP, what you can see is that you basically increase your false positives. And so your specificity decreases and your positive predictive value also decreases. So at least at Merck, we don't routinely look at MRP2, 3, and 4 inhibition. So with that, we'll dive into our second in vitro system. So when assessing the potential for transporter-mediated cholestasis, testing inhibition of each transporter in isolation in vitro may provide a limited view of what occurs in vivo, right? Using an hepatocyte-based system may be a more advantageous approach to examining the effects of drugs on bile salt transport, because you can take into account things like metabolism, protein binding, intracellular sequestration, or compensatory transport mechanisms. Now, HepatoPack is a bioengineered microliver platform, which can serve as a functional model of the liver in vivo. Micropattern plates contain tiny colonies of organized hepatocytes surrounded by supportive 323 fibroblasts. This is just some characterization data showing fluorescent microscopy of the HepatoPack system. So the figure to the left-hand side shows the presence of intact biocannuliculi by looking at CDF dye. So this dye actually diffuses into the hepatocyte. The dye is cleaved, and so that cleaved dye is then a substrate of MRP2. MRP2 will pump it out into the biocannular space, and that's the image that you're viewing. So we can form biocannuliculi in this system. This data also shows that MRP2 at least is functional in this system. To the right-hand side, what you're looking at is just localization of BSEP and MRP2 on the cannulicular membrane, right? So we know BSEP is localized. I'd like to take a few minutes to walk you through a typical transporter experiment in the HepatoPack system. So cryopreserved human hepatocytes and stromal cells are plated in cold culture, and hepatocytes are given time to form tight biocannuliculi. The cells are then pre-incubated with inhibitor in buffer, either containing or devoid of calcium for a given amount of time. The cells are then washed and incubated in a buffer containing a radio-labeled substrate. In this case, we're using TCA, plus or minus inhibitors, and then your cells are washed, lysed, and counted by liquid scintillation counting. Now, the key to the study is really the presence or absence of calcium in the buffer. And so in the presence of calcium, what happens is you maintain the tight junctions and maintain the formation of your biocannulicular space. So if you examine the uptake and the efflux of taurocholate will be taken up into the hepatocyte, mainly by NTCP. It'll be effluxed into this cannulicular space by BSEP, and then it will just sit there, right? Because the biocannuliculi are well formed, we have tight junctions, it will just remain there. So if we lyse cells under this condition, what you're looking at is the amount of taurocholate in both the cells plus the bile. Now, in the absence of calcium, you don't maintain these tight junctions, and these biocannulicular spaces are disrupted. So if you look at the uptake and efflux of taurocholate will be taken up into the cell, and then it will be effluxed by BSEP. It's still sitting on that membrane, but whatever's efflux will just leak out into the media. So if you lyse cells under this condition, you're just looking at what's in the cell. So if you know what's in the cells in the bile, and then you know what's in the cells, you can do some simple subtraction to find out what's in the bile. And from an experiment like this, you can calculate two important parameters. The first parameter is BEI, or biliary excretion index. This represents efflux across the cannulicular membrane. And so to calculate this, you look at your accumulation in cells plus bile minus your accumulation of cells, which just leaves you what's in the bile, divided by your total, or the accumulation in cells plus bile. Now, the point I really want you to remember from this slide is that if BEI represents efflux across the cannulicular membrane of the hepatocyte, and BSEP sits on the canalicular membrane, and efflux is compound across that membrane. If you inhibit BSEP, you should be decreasing BEI in this more holistic system, right? So remember that. The other important parameter we can calculate is the in vitro biliary clearance, and this represents the net efflux of uptake and efflux into the biocanalicular space. So once we calculate these parameters, we can start to interpret the data that we're getting out of these experiments. So I told you in vitro biliary clearance represents both the uptake and the efflux, and BEI is just the efflux component. So if we inhibit in vitro biliary clearance without BEI, we can conclude that the cytosolic uptake pathway is affected. If we inhibit both parameters, we can say the uptake and the efflux pathways are affected. If we don't inhibit in vitro biliary clearance, but inhibit the BEI, it's just the canalicular efflux pathway that's affected. So we can use this to now ask the question whether known inhibitors of BSEP, right, in the vesicular assay cause a decrease in BEI in this more holistic system. So we have two experimental designs with two pretreatment times. So in the first one, we have a 10-minute pretreatment with buffer plus or minus calcium in the presence of absence of potential inhibitors. We pre-treat for 10 minutes. That's long enough to disrupt these biocanalicular spaces. And then at time 0, we add our TCA plus or minus our inhibitor. We wait 10 minutes. This is in the linear phase of the uptake and the efflux. And then we lyse and count. Our second pretreatment time point is a 24-hour time point. We added this primarily to make sure that we were able to incorporate the effects of potential metabolites that are formed in this system. So, for 24 hours before the start of the experiment, we pre-treat in serum-free media, plus or minus potential inhibitors. And then 10 minutes before, we're just adopting our workflow up here. So 10 minutes before, we pre-treat plus or minus calcium buffer in the presence or absence of potential inhibitors. We add our substrate at time 0, plus or minus potential inhibitors, and then we lyse and count. And through all this, we calculate the in vitro biliary clearance and the BEI for both the 10-minute and 24-hour pretreatment. The cells in the supernate are collected after a 24-hour pretreatment for MetID analysis. So we want to understand what's happening to our compound and whether metabolites are formed. And then we also run a cytotoxicity assay for the 24-hour time point, just to ensure that any changes in the BEI that we observe are not due to toxicity. So this is what the data looks like. What I'm showing here is just a positive control cyclosporin. If you start on the left-hand side of the slide, you're just looking at DPMs per microgram of hepatic protein, and you're looking on the x-axis at increasing concentrations of cyclosporin A. And what you can see is a concentration-dependent decrease in the accumulation inside the hepatocyte in the biliary space in the presence of calcium or in the absence of calcium. At each one of these concentrations, you can calculate in vitro biliary clearance or BEI, right? You can normalize to percent control, where the in vitro biliary clearance or the BEI in the absence of test compound is defined as 100%. You can plot percent control versus increasing concentration of cyclosporin, and you can generate an IC50. So for in vitro biliary clearance, you can see here that the IC50 was 2.4. For BEI, the IC50 was 4.7. And I told you previously that if you inhibit both in vitro biliary clearance and BEI, that you're affecting both cytosolic uptake and canalicular efflux. So to help put data coming out of this assay into perspective, what we did was we took a subset of the 121 DILI positive and negative compounds. So we took 30 compounds from the 121, 19 were DILI positive and 11 were DILI negative. And we generated BEI IC50. And what I'm showing you here is 1 over the 24-hour BEI IC50 on this y-axis. And again, on the x-axis, we're just looking at the unbound liver inlet concentration. Again, green is DILI negative compounds, red is DILI positive compounds. This red dotted line across this graph represents a 10-fold difference between 1 over the IC50 and the unbound inlet concentration. Compounds that fall to the right of the line have IC50s within 10-fold of that unbound inlet. So those would be compounds that we would be concerned that perhaps in vivo they would be able to inhibit BSEP, where compounds to the left-hand side would have IC50s greater than tenfold. And so we would conclude that these have a less chance of inhibiting BSEP in vivo. This black line down here just represents cases where the IC50 was greater than the highest concentration tested in the assay. And so since the IC50s were unknown, it cannot be compared to the free inlet for compounds that fall to the right side of the line. So these compounds on this side right here weren't included in our sensitivity analysis because we can't make a judgment as to whether or not these are actually positive or negative because we couldn't test them high enough. Looking at this assay, we had high specificity, so we had one false positive. Again, our sensitivity is low, which is not necessarily surprising. So you have red X's on this side of the line. Again, it could just be due to a different mechanism. But I think what's really important to highlight here is that compounds that I just circled in black. So 9 out of the 15 compounds, these compounds were flagged in vesicles, right? But they did not inhibit BEI such that the IC50 was within tenfold of the free inlet concentration. So in vesicles, all the compounds that are circled, we would have highlighted as potential risk. We can take it in this assay and we can see that 9 of them, right, actually really have low risk, including our false positive in the vesicular experiment, dipyrimidol. It was shown that this compound actually was metabolized, and that's probably the reason for the false positive in the vesicles. So we're able to utilize this as a downstream assay from a vesicular assay to further define risk. So let's go back to MK866. That was a compound that had the IC50 of 0.8 in vesicles. What you can see from this table is that it did inhibit in vitro barrier clearance to some degree. Didn't do much around the BEI. But when you look at the free inlet concentration, you can see clearly that there's definitely a greater than tenfold window between the IC50 for in vitro barrier clearance, or BEI, compared to that free inlet. And so thus we would conclude that the risk of a clinically meaningful in vivo interaction with BSEP was low. And actually this turned out to be the case for this compound. So we also had clinical data to support that showed there was no increases in plasma bile acid levels in the clinic. And a colleague showed that it was actually likely that a reactive metabolite was causing the issue in the clinic. We wanted to take this one step further and just kind of understand the discrepancy between the vesicular and the hepatopac assay. What we found was that approximately 80% of MK866 was converted to an acyl-gluc metabolite following a 24-hour pre-incubation. We actually made this acyl-glucuronide. We tested it in the vesicles, and the IC50 was found to be greater than 25. So the metabolism of MK866 in a hepatopac may explain the discrepancy between the two different assays. In addition, high protein binding of MK86 could also contribute to the lack of inhibition in hepatocytes. So I'd like to show you what we learned when we merged these two in vitro assays together. So what you're looking at here, again, are just assay results from our BSEP vesicular inhibition assay, or our hepatopac assay. Green is a negative result, right? So in the vesicular assay, this would mean that your IC50 was greater than 5 micromolar. You were below that empirical cutoff. Red is a positive, so you'd be above that empirical cutoff in the vesicles, or you'd be within 10-fold of the free inlet in hepatocytes. And you're looking at those results in DILI negative and DILI positive compounds. These are the compounds that have been through both assays. And what you can see is that inhibition of BSEP in the vesicular inhibition assay may be predictive of DILI risk. And so you see all the red assay results, or the positive assay results from DILI positive compounds in a vesicular assay, and our one false positive that was DILI negative. But of course, causality has not been demonstrated. The other thing I think you can see is that inhibition of BSEP in our vesicles is not always predictive of inhibition of canurical efflux of TCA in a hepatopac. So you can see positive DILI compounds that are positive in our vesicular assay, and when we put them in our hepatopac, we're able to further de-risk them. These discrepancies may be explained by metabolism, protein binding, and or compensatory mechanisms. So caution is warranted in interpretation of the vesicular inhibition data in isolation. Finally, I want to wrap up with a few conclusions. So the International Transporter Consortium recent BSEP white paper recommended the proactive evaluation and understanding of BSEP inhibition to aid internal decision-making on potential human DILI risk. This two-tiered in vitro approach can help de-risk drug candidates from potential BSEP inhibition liabilities and limit compounds with major liabilities from reaching the clinic undetected. However, there are caveats to all approaches, and gaps in our understanding of BSEP-mediated cholestasis remain. I'd just like to highlight for a second some of the caveats. So I think in any kind of decision tree approach, you're willing to take on some risk, and we are as well. Some of the caveats here are that only test compounds which are potent in the vesicles, right, get tested in hepatopac. If you weren't potent in the vesicles, but potentially had a metabolite that inhibited BSEP, we would miss that compound. So we haven't seen that. So all the DILI negative compounds that we tested in hepatopac from our test set that I showed you before, we only had one false positive. While we don't routinely screen compounds that are not potent in vesicles, we do run some issue-driven studies from time to time, and we haven't seen a compound that was negative in the vesicles be positive in hepatopac. But maybe, perhaps we've just been lucky, and it's certainly something that we could miss. So that's one caveat. The other caveat is that BSEP inhibition does not necessarily lead to cholestasis, right? And so there is potential to take a compound and stop developing it, right? Because of this liability, along with maybe some other liabilities that the compound has, and the compound may actually never be cholestatic in the clinic. So those are two of the caveats. I'll just say that since the induction of this workflow, several years ago, we've had no issues with BSEP inhibition after implementation of the workflow, and it really helped us reduce liabilities from reaching the clinic. Finally, I'd like to just say that our current efforts are aimed at better understanding potential of preclinical models to assess BSEP inhibition, as well as explore approaches to translate findings to humans. So I'd like to finish just acknowledging my colleagues across the Merck organization, from my department, PPDM, from the safety organization, as well as the genetics and pharmacogenomics groups, particularly Raymond Evers and Frank Sestaire for their insight in developing this strategy, as well as colleagues from BioIVT who helped run the hepatopac studies. And with that, thank you for your attention, and I'll turn it back over to Kaushik.
Excellent, Mike. Thank you very much for that really nice summary of how BSEP may interact with may play a role in drug-induced liver injury. Moving on to the next topic is sort of a change of gears, because we talked about this briefly at our last meeting. This was in October, when Kevin Koh talked about reactive metabolism and how formation of a reactive metabolite leads on to modifying proteins that then can be a cause for drug-induced liver injury as well. So for the last 2 decades almost, we have looked at drug metabolism and reactive metabolism in particular, using tools like trapping studies with GSH or cyanide or semicarbazide or covalent binding corrected for dose, perhaps, to assess risk for drug-induced liver injury. What Jim and Wen are going to talk about is perhaps the next stage of assessment of drug-induced liver injury through bioactivation, where they are going to give an overview of a transcriptomics analysis, something that is perhaps a cellular readout of benign versus dangerous adducts. And so what I would like to do first is introduce Jim, who is going to talk about the work that he has done at Merck in the last decade or so, working on generating a transcriptomic signature for in vivo in the rat. Jim is a senior principal scientist in the investigative toxicology group within safety assessment and laboratory and animal resources at Merck. He has led efforts to develop novel in vitro and in vivo models in the entire time there at Merck as a mechanistic problem-solving tool for supporting several different kinds of risk assessment and lately in the DILI space. Jim received his PhD in biological chemistry and molecular pharmacology from Harvard and did his postdoctoral research in epigenetic toxicology at Merck Research Labs. Over to Jim.
Good afternoon, everyone. Can everyone hear me and see the slides?
Yes.
Terrific. So what I'm going to share with you today is a rat liver transcriptional biomarker, a platform that we call the bioactivation liver response assay that we apply early in lead optimization for mitigating risk of clinical drug-induced liver injury. And so I'm going to use a few abbreviations, BA-LRA to refer to the assay, DILI as an abbreviation for drug-induced liver injury, and I'll talk about DILI drugs. And so I think the final point to make here is that this work has been recently published, and I include a link for you to go to that paper and see some of the details that I won't have time to share today. So what I'm showing you in this first slide is some of the rationale for why we made this investment. To the left are meta-analyses from the literature that describe the fact that liver is a major target for drug attrition and market withdrawals. So in the upper left, the attrition and the contribution of the liver for attrition for drugs in clinical development. Here in the lower left, drug withdrawals, and again, the significant contribution of the liver to those events. And here on the right, just a snapshot of preclinical events that were cause for discontinuation, and again, seeing the prominence of liver in those events. You know, the bottom line here is that human and animal toxicities are not perfectly concordant, and so the use of preclinical species for anticipating these types of injuries are often met with failure, and so there's a need for new assays, particularly when one is trying to avoid these late-stage discontinuations that are both compromising to the patients that are exposed to these potentially harmful drugs.
Can I interrupt you for a second, Jim? There seems to be a couple comments that folks are not being able to see your slides.
Okay, who can help me with that?
David?
We'll respond to these people via text, but thanks for bringing it up.
Okay, great, thank you. Good to go, Jim.
Sounds good. So yeah, so again, the main goal here is to come up with better models that anticipate these clinical events earlier and avoid exposing patients to adverse drugs, and that minimize the lost investment on the part of drug sponsors. As Kaushik alluded to in the introduction, the chemical nature of reactive metabolites was for a long time a way of trying to de-risk for DILI. The ultimate outcome of that type of work was that just the pure chemical reactivity measures are not robust in terms of their predictive value. I'm showing you here results from a paper by Nakayama et al. And in the left panel, you can see how just the raw covalent binding numbers do not differentiate between drugs that are safe in blue, drugs that are associated with liver warnings in yellow, and drugs that were actually withdrawn or that received black box warnings. And what was interesting about this paper is that they applied this concept of body burden. So looking at these data in the context of the daily dose of the drug, and there one began to see some differentiation of these classes where one could make a distinction. So it wasn't just the absolute amount of covalent binding, but actually that multiplied by the dose of the drug. Our experience within Merck was not as promising. So we tried to recapitulate these types of experiments shortly after this paper was published, and so you can see here safe drugs for DILI in green, adverse drugs for DILI in red. And while the sensitivity of this assay for DILI drugs was perhaps encouraging, what we felt was that the specificity was below a level that we found acceptable. Too many good drugs were being sort of thrown out. And so I think the point that was made earlier, that the real challenge to resolving and de-risking DILI risk is so multivariate. There's so many sources of DILI mechanisms that we shouldn't expect any one assay to solve our problems. And so you've heard Mike talk about the transporter assays. You're going to hear about I'm telling you about the in vivo, and you'll hear Wen tell you about the in vitro reactive metabolite assays. But the point I want to really drive home here is that any given assay may appear to be fairly modest in terms of its sensitivity for a broad spectrum of DILI drugs, but what we cannot overlook is the specificity. That remains paramount, even if we can tolerate some compromise in terms of the level of sensitivity. And so, as I said, these are platforms that we are integrating into our work process very early in lead optimization. At Merck, we conduct a very early study for early candidates called the Safety Lead Optimization Study. This is a resource-sparing rat study. It's 5 days. We get quite a bit of useful information in terms of in-life endpoints and some understanding of potential toxicities. We focus on 4 major organs that are kind of high-frequency hits for problems in moving compounds forward. And what we had done up to the time that we came to developing the BALRA was developed transcriptional markers that could diagnose toxicity in these organs. And so with this backdrop, what we started to ask ourselves was, could we identify transcriptional signatures of the bioactivation phenotype that would allow us to also begin to de-risk the formation of chemical reactive metabolites that had risk? So the reason to think about that, and the kind of the hypothetical construct around that, I try to outline for you here. One can think of the reactive metabolite being formed in the body, and one can think of its sort of naked chemical reactivity impacting macromolecules in a cell directly. But we also began to think about the potential for kind of defense pathways activating in a cell, in a hepatocyte. How might the hepatocyte respond to such insults? And the well-recognized activation of NRF2 pathways and the activation of genes that mount defense to electrophilic and oxidative stress were sort of the goal of these studies. And so where we started was to actually employ some of the historical knowledge that we had about covalent protein binding at Merck. And so we identified a series of molecules that we would have defined based on the parameters at the time as having a high covalent binding characteristic all the way to a low covalent binding characteristic. And we administered these to rats in the slow study paradigm. So what you can see in this heat map is that we were clearly able to identify a set of genes that correlated well in terms of their upregulation in response to those drugs that had high and medium protein covalent binding, and that we could in fact differentiate those classes, those risk classes of compounds from drugs with perhaps a low or what might have at one time been considered a safe level of reactivity. What you can see in this panel here is that if one set a threshold of a BA-LRA score, which was essentially defined as a log transformation of the average fold change across the genes, a sum of that, you could see that we could generate fairly reasonable sensitivity and specificity on the order of 70% or so, again, employing that threshold. So this gave us some optimism, if you will, that we could potentially apply this to drugs that had a history of DILI phenotype in the clinic, and that we might be able to pick out those that perhaps occurred through a mechanism that involved bioactivation. From these studies, we empirically understood as well that the dose of the drug administered to the rat was important. And so in this last panel, what you can see is that the behavior of, or if you will, the dose response of these BA-LRA scores was definitely associated with the dose administered to the rat. And so we understood that doses below 300 mg per kg per day would not reveal all of the potential culprits, and that we actually had to push dose in these studies. And so this began to modify the study design of the BA-LRA, where we started pushing dose in order to uncover these molecules that harbored the reactive characteristic. So the next step then was to bring in a test set of drugs that actually had a DILI phenotype. And what I want to just say up front is that these drugs were selected independently of what their potential mechanism of DILI may have been. So we were agnostic to what might be known about a molecular mechanism of DILI from individual drugs, and solely focused on their clinical phenotype. And ultimately, we would go on to test on the order of 130 or so drugs that had been curated for DILI clinical phenotype or liver safe clinical phenotype. And I can show you the outcome of those studies here in this slide. So again, borrowing from the Nakayama treatment of the data, we essentially replaced the covalent protein binding with the BA-LRA score, and again, safe drugs in green, red adverse drugs in the red X's, and ultimately what we were able to achieve was a sensitivity of 32%, but again, which might seem modest, but again, maintaining that very high specificity, which meant that we were not throwing good drugs into the trash bin. And so, back to this question of the level of sensitivity, remember that 100% of the DILI drugs that were put into this metric, into this analysis, were injurious to people. And so, when you look at it through that lens, the 32% may not seem so insignificant. And I think that the fact that we're able to maintain the fidelity for not calling out what are ultimately safe drugs is an important thing to consider. So where the rubber meets the road, or where you start to scratch your head, is really trying to understand this lower quadrant here, where you've got this sort of Christmas tree lights assortment of red and green, and you start to ask yourself, okay, why are there so many false negatives, so to speak? Why are there so many red X's down there? And so I think you can theoretically suggest a number of possibilities. Maybe these aren't drugs that cause DILI through a reactive metabolite mechanism. And so this is the wrong metric, if you will, for assessing them. We clearly identified examples of molecules that, and I think Wen will describe an example of this for you in the case studies that she covers, where metabolism between the rat and the human are different. And so, you know, that is an assumption that goes in, that the animal model will reflect the metabolism in the human. But then a third possibility that one has to consider is, you know, did we meet a sufficient exposure metric? Did we deliver sufficient drug to the liver in order that the chemically reactive metabolite was at sufficient levels to trigger the defensive response of the hepatocyte to the electrophilic stress? And so in that vein, we started thinking more carefully about, well, what would be the appropriate exposure metric? You know, typically we look at peripheral exposure in toxicology studies to establish margins and understand, you know, the level of concern about a given finding with respect to you know, what doses one will ultimately deliver in the clinic. So we started to ask this question, and what I'm showing you here, what I want to explain to you in this slide is an empirical measure that we developed in the group to understand what would be a putative threshold. And this is still a work in progress, but this is a snapshot of roughly 150 molecules that were part of our early lead up characterization. And so each individual dot represents an individual molecule. And what you can see is that they're different colors, and those colors represent the peripheral exposure that was achieved in those studies, 50 micromole hour and below, between 50 and 200 micromole hour, and 1,000 or greater. And I think what you see right away, if you look here in the upper right-hand quadrant, right, this is the quadrant with the molecules of concern, if you will, where you're above the 0.22 threshold. And what you can see is that there is a mixture of all these different colors. In other words, peripheral exposure does not correlate very well with a BA-LRA score of concern. However, if one begins to consider a local concentration of the drug, in other words, if you start to think about how much drug made it to the liver, there you can begin to construct a dose-response relationship and contextualize, if you will, almost a potency of how much of parent drug made it to the mouth of the liver, made it to the portal circulation, and then resulted in some score. And so to do that, we worked with our colleagues in PPDM, we worked with our colleagues in PPDM to develop this so-called estimated absorbed dose. And so what this reflects is the fraction of drug that escaped the gut metabolism, the fraction that was absorbed, the fraction that escaped the gut metabolism and made it to the portal vein. And this is something that is calculated, it's not measured, but one can get a fairly good estimate with certain assumptions, with data from very routine IV and oral dose toxicokinetic parameters. And so what you see, in contrast to the peripheral exposure measurements, is that as you begin to push out in terms of the estimated absorbed dose, more and more of these molecules are revealing their capacity for a significant BA-LRA score. And so we started applying this metric as a threshold for determining when a molecule had been sufficiently tested for trusting in the BA-LRA measurement. There are a couple additional figures in this representation of compounds, just for good measure to sort of drive home the point. These red squares represent molecules that, again, had very high peripheral exposures. But what when we drill down into these molecules and understand better their physical chemical properties, what we understand is that they have very high levels of protein binding. They're long-lived in the circulation, and they have very low clearance. And so, the apparent high exposure peripherally for these molecules, but clearly their lack of turnover because of these physical chemical properties would have fooled us into thinking that these would have been negatives that were BA-LRA negatives that had been adequately tested, when in fact they just weren't properly absorbed. By contrast, these green diamonds represent molecules that had very high intrinsic clearance, and so they would have been turned over very fast. And so what you can infer from where I'm taking the argument is that these are the sorts of molecules that you really feel safe, that had good exposure to the liver, had good turnover, had the opportunity to generate metabolites. And so these two diamonds above the threshold, these are molecules that you feel are really soundly tested, and you're now going to consider these and their score in the context of the clinical dose that you would administer for whether or not this is a signal of concern. So I'd like to just take a moment to describe a couple of case studies. I'm showing you results for the BA-LRA score for a historical compound in a program at Merck. So against the backdrop of our tuning fork, if you will, that 130 compounds and the relevant thresholds, you can see that this compound in red, compound A, scored such that compounds, a dose of above 300 roughly in a human would put you into this quadrant of concern. You would sort of join this group of molecules that have the adverse finding clinically and the elevated LRA score. And so what's really interesting is that when you look at the clinical historical data, those data fit very nicely with the prediction from this body burden treatment of the BA-LRA score. In other words, doses above 300 or so, you expect to have some clinical event that's adverse. That's consistent with the increased frequency of patients at doses greater than 500 milligrams in the clinic for this molecule. And perhaps doses below, say, 200, 100, you'd expect a low frequency of adverse events. And as you can see, that is borne out by the true clinical outcomes that we're seeing for this molecule. So this now became a way for us to go back and look at backup molecules and try to dial out in an SAR-driven fashion the features of compound A, and that resulted in the identification of molecules that, all other things being equal, we were able to dial out the LRA liability and identified compounds B and C. Ultimately, this program was not continued at Merck, but compounds B and C were out licensed, and we got really good news very recently that compound B had made it through clinical trials. It recently received approval for marketing with no liver safety label precautions. And so this is just the idealized case of how we apply the BA-LRA to identify molecules with reduced risk and move forward the best candidates into the development space. The second case study, what I want to emphasize here is how, and I think Mike emphasized this really nicely, how these assays really aren't meant to be go, no, go decision makers. It's really a weight of evidence approach. And we worked very closely with our colleagues in PPDM and our colleagues in chemistry to not only generate these red flags, if you will, but also to learn something about the nature of the structural risk that's contributing to the LRA score. And so what you can see here are schematics of molecules and kind of the structural elements that were ultimately identified as being responsible for, were sort of the reactive hotspots. And what you can see is that we went through these two molecules in the rat LRA study, and we had these positive LRA findings. Rather than iteratively run these, albeit resource sparing, but still, studies that are slow in turnover and not high throughput, we decided to start interrogating those molecules and try to identify trapping assays from our PPDM colleagues that might correlate with the positive finding. And sure enough, the semicarbazide trapping studies nicely aligned with the risk that was identified in the LRA. So one can see here now a schematic of the structural ideology, if you will, understanding that semicarbazide could identify these reactive metabolites, and we could now begin to formulate some hypotheses about where chemists might look for the next iteration of these molecules. And so additional molecules were identified that were thought to not have the same risk. These were run in the trapping assays, and happily they were shown to be relatively clean in terms of the semicarbazide property. So this was now showing us differentiation, structural differentiation. So a rational based approach to differentiating this series, and ultimately this correlated very nicely with subsequent in vivo LRA studies that were conducted. So again, just to draw the point a little bit more clearly, the goal here is to work very closely with other assay platforms, other subject matter experts to really define a rational drug design approach that mitigates the reactive metabolite risk. So I'm going to leave you there with a few conclusions. I hope I've convinced you that DILI is a major contributor to attrition, and that it's incumbent on us to identify these problems as early as possible. This slow paradigm, again, relatively resource-sparing animal study, we can use it in a number of ways to identify important development-enabling endpoints, but that these studies have been effectively leveraged for identifying reactive metabolite formation and the risk associated with DILI, and that we work with our colleagues to not only identify the hazard, but to identify what are the structural bases for those red flags. Ultimately, as has been stated, DILI is not a one-size-fits-all. There are a number of other mechanistic sources of injury in the clinic that we continue to work toward identifying appropriate assays. And so this really is a continuous refinement, not only of the existing platforms, but also a search for new platforms. And so I'm going to leave you with acknowledgments for all of my colleagues that contributed to this work, past and present members of the investigative tox group, the pharmacogenomics group, drug metabolism, chemistry, and of course our terrific tox and path groups that do such a great job of supporting us. And with that, I will pause and hand it over to Kaushik.
Excellent. Thank you, Jim. I am going to do a quick introduction for Wen for the transition from the in vivo to how Wen and her group has been able to use this in vitro early on in the discovery. Wen is the director of Genetic and In Vitro Cell Toxicology Group within Safety Assessment and Laboratory Animal Resources at Merck. One of the focused areas of her group is the development of new in vitro assays and endpoints, one of which she is going to talk about today. And what's interesting also is that the problem-solving tools that they come up sort of as a response to a finding to help reduce those issues for compounds that are backups, for example. Wen also serves as the discovery program rep and responsible for program-related non-clinical risk assessment strategies. Prior to joining Merck, Wen was a research scientist at Applied Genetic Technologies Corporation, and her PhD is from the University of South Carolina and a postdoc in virology at the University of Pennsylvania. Wen?
Thank you, Kaushik. Can you hear me?
Yes.
All right. Thank you, Kaushik, and the organizer for the invitation for us to talk to you about some of the work we have done in the past 6-7 years to develop a transcriptomic-based approach in the advanced in vitro liver model to reduce the spike activation potential and drug-induced liver injury. I think I'm gonna cover like the business need, but I realize Mike and Jim has covered really well, so we'll be brief. So I think my main presentation will be focused on showcase some of the examples that we feel the in vitro transcriptomic-based bioactivation signature, whole that signature based approach can help in the early drug discovery space to reduce bioactivation potential and daily risk. I'm going to give you some of the work we are doing with the human hepatopac and how in some cases the human hepatopac can help us to gain more perhaps human relevant risk assessment. So I'm not going to actually go into details because Jim did a really nice job tell you about the bioactivation, the risk factor for DILI, and the traditional method of measuring bioactivation potential, right? So Jim and it was in Merck and as well as a number of companies had actually leveraged this red liver gene expression biomarker approach to perhaps, if you think about it quantitatively, measure the bioactivation potential in the preclinical species, then help to actually conduct risk assessment in a preclinical space. So what our group has worked on is really try to translate that in vivo biomarker response to an in vitro platform. As many of you guys online or chemists, as you are fully aware, once we identify the risk, there's an urgent need for us to come up with in vitro assays so quickly enable our medicinal chemists to look at the backup and test SAR-based hypothesis, right? So obviously the advantage is, we could only, say for the in vivo study, you may need a gram of compound, but the in vitro study, you only need a milligram quantitative of compound. You can screen a large number of compound all at once. I think also has a potential if you think about the cross-species translation, right? Perhaps we can get to the point in the future that we use the in vitro human-based models to help us de-risk or understand the human risk. So I think Mike has described the hepatopac platform very nicely. So I'm not gonna go into the details. So we have looked at a number of in vitro liver platforms at the very beginning of this effort, and we focus on a number of key parameters that we feel are important for us to have the confidence that the in vitro response we see with the in vitro liver model will be closely mimicked the in vivo response. And in the early discovery space, obviously, we also want to have a platform that's robust and reproducible that can be easily implemented in the early discovery space. So this is just showing some of the key properties we considered that's important to decide on the in vitro liver platform. Some of the published literature clearly demonstrate the HepatoPac platform show stable and long-term metabolic capability compared to, say, the monolayer culture of hepatocytes. And recently, the group in Roche published a very nice paper. They look at various liver platforms, very comprehensive study to look for really the differences in terms of the performance when it comes to drug metabolism. As you can see, the HepatoPac seems to perform really well and have like high metabolic activity, fairly comparable to a fresh hepatocytes. And we actually pay a lot of attention to the biochemical synthetic pathways or hepatocyte function, as we know, when they exist in vivo in the liver. So we look at surrogate as that function using albumin and urea production rate in vitro liver platforms. Here, just showing you albumin and urea production rate over a period of 2 weeks. So we looked at the rat hepatopac, we looked at human hepatopac from individual donors, and then we compare that number to what we estimated in the literature estimate what the in vivo production rate would be. And as you can see, the HepatoPac platform really produced numbers fairly close to in vivo liver, rat or human. And over the 2 weeks period we evaluated, it seems to have fairly stable production rate as well. What we also did is to look at the transcriptional profiles of the HepatoPac platform compared to in vivo liver. This graph on the left basically look at human hepatopac versus human liver. As you can see, the hepatopac platform, really, if you look at the global gene expression, they're fairly close to the human liver, and they are fairly stable. They don't have that drift as you would see with some of the other platforms, either iPSC derived or the primary hepatocytes cultured in monolayer. So that tells us really, the platform is fairly similar to hepatocytes in vivo setting. Similarly in the rat, although in the rat, perhaps if you look at the principal component 2, which only count a small portion of the analysis, there's some room for improvement for the rat hepatopac. But if you look at principal component 1, which is the majority of the analysis, they're fairly close to the liver, and they seems to be fairly stable. They don't have that large drift that what you would see in the with the primary hepatocytes. So with that, we feel the HepatoPac is a really good platform for us to develop that bioactivation gene signature. So we designed study to mimic what we do in the rat in vivo study Jim just described. So we end up adopting a design. We use repeat dosing studies, so the cells are treated every other day for a total of 9 days. So we collect endpoint, intra-endpoint for measurement of cell viability, and occasionally we do metabolome profiles as well as metabolomics, look at endogenous metabolites through those median endpoint. At the end of the study, we'll collect the cells, run gene expression analysis with a relatively high throughput mid-density array platform. Just very similar to what Mike and Jim described. So we look at about over 90 compound that's either considered positive for drug-induced liver injury or negative. Here showing green and red, and we see, clearly there's a lot of DILI positive compounds show a very nice dose response. And what we also did, similar to what Mike described earlier, is to put that response in the context of relevant clinical exposure. Here, similarly, we use the liver inlet, calculate liver inlet concentration. So we use this liver exposure index, which is just the test concentration divided by what calculate to be the total liver inlet concentration. So as you can see, the positive drugs actually, a majority of them has a pretty robust response at the liver exposure index, like lower than 3. Well, we do have some false positives for the DILI negative drugs, but majority of them either did not have a response or have a response at a very high liver exposure index. So overall, the assay is performing relatively well for us. With the qualification test set, we have greater than 80% sensitivity and specificity. Just quickly to show you some of the examples, we feel the assay is useful. First example is, like if you think about the lead ID and early lead up space, when chemists want to look at different structure class, whether there are intrinsic liabilities for bioactivation and DILI, and you can use assay to help you. This is using some of the well-known DILI positive and negative drugs to demonstrate that principle. As you can see, the bosentan and cisexaten in this in vitro red liver, in vitro hepatopac liver response assays, you see clearly very positive response. While the negative drugs, we did not observe a significant response. When we use the liver index metrics, that clearly separate this drug into this quadrant of being high risk and this lower quadrants of being safe. I think a lot of the utility of this assay has really been helping our chemists to look at SARs to help them test some of the hypotheses they developed experimentally. Here, just showing you an example again with some marketed drugs, clozapine and olanzapine. These drugs are fairly close in structure. Obviously, clozapine has a high risk for clinical DILI. As you can see, when we test them in the rat hepatopac LRA, clozapine has a very robust response compared to olanzapine. I think what we want to kind of, show here also, if you think about the traditional method of measuring bioactivation right here, and show you if you look at covalent binding or GSH adduct or TDI, these two drugs really kind of hard to differentiate, right? So when we have data like this, it's really hard to, I think with this in vitro transcriptome-based assay, perhaps you can think, this may give you that outcome like Kaushik mentioned earlier, right? It's a bad adduct versus maybe not so bad adduct, or a balance of your reactive metabolism pathways versus some of the detox pathways. Just quickly, so obviously not all drugs can be differentiated by this assay, just like what Jim said earlier. For example, if you look at triglyceride and rosiglitazone in this assay, we see very similar response in terms of concentration-based response. But if you say, you can differentiate them with the liver exposure margin, that's clearly, you can do that. I think that kind of bring the point is really important when you look at the in vitro assay results, and it really have to be, thinking about the potency and the exposure to give you a more like weight of evidence approach to have the proper risk assessment. So we did some of the mechanistic studies with ABT or SRA to block P450 mediated metabolisms. Here, just show you acetaminophen obviously have a very strong response in the rat hepatic LRA, but if you block P450 with either SRA knockdown cofactors or the ABT, you can abolish or greatly reduce that response. Human hepatopac, this is something we are actively working on. So we did limited evaluation with a single human donor hepatopac, and you can see for this I'm showing here, this single donor hepatopac, obviously similarly with this transcriptional response, it performed pretty well, perhaps a little bit less than what the rat hepatopac in vitro assay performed. What we did notice is there's quite a bit of donor-to-donor variation for the human hepatopac because we're using single human donor lot. That's not surprising, perhaps, but we are kind of in the process of understand, how important it is to understand that donor-to-donor variability and how important it is to have something with a pool donor, perhaps have a more consistent response. I think I'm just going to go really quick here. The human hepatopac, what we feel can bring really value is when you have metabolism differences, right? As bowel activation is metabolism mediated, when you know the potential for that difference, really, you ask yourself whether you should employ the human rather, in combination with the rat to help you to actually perform more human relevant risk assessment. Here, giving an example of clopidogrel. Clopidogrel is a drug that's well known for reactive metabolite formation. As a matter of fact, the pharmacology actually is through reactive metabolite, right? So consistent with all those findings, and our in vivo LRA assay suggests this drug will be associated with a daily risk at perhaps what the dose that's being used clinically. I think the question we ask ourselves is, is clopidogrel not really associated with high incidence of DILI, although there are rare cases. Well, we have all this data suggests a reactive metabolite liability, and how do we reconcile that? I think that perhaps, if you look at the human hepatopac and the rat hepatic data, that perhaps provides the answer. If you look at the graph on the right for the human hepatopac, we really did not see a really robust response up to the 100 micromolar, but in the rat, we saw a pretty robust response. And we took this median, look at MetID. I think you can see the human, really, the parent turnover is high, with majority of the metabolite being the hydrolysis product. Well, in the rat, the turnover clearly is lower than in the human, and we also see significant amount of oxidative metabolite. So what we think is happening is really probably in the rat, the clopidogrel is driven through to more higher degree through the P450 mediated metabolism that created reactive metabolite activation consequences. Well, in human, the hydrolysis pathway is probably more prominent. So we test that with metabolite taken from either parent or the P450 metabolite or the hydrolysis metabolite. As you can see that, the strong response with the parent and the cell lactone seems to confirm our hypothesis. Overall, I think with this very quick presentation, that I hope I show you that this in vitro transcriptomic-based approach can be used to help you de-risk chemical entities that find drug candidate with lower bioactivation potential and delay risk. And we have seen pretty good performance with the rat hepatopac platforms. the human, we have limited data suggests perhaps we can develop something similar, although with a single donor, we do see a slightly lower predictivity. And right now we, use the rat as our main work horse, and that the human obviously used on a case by case basis if strong rationale exists. I think, overall feel this transcriptomic-based approach in vitro can be useful to help early chemistry effort to test the SAR hypothesis and help the candidate selection. Yeah, I think there's a lot of people involved in this work within Merck, as well as our colleagues at BioAD. I think that's my last slide.
Excellent. We are at 3:30, but I think we had an extension for a few minutes to answer some of the questions that we have. The majority of the questions are around the transporter work, so I'm going to get started on those. There is a question around for Mike. Are there any fragments or functional groups that could be avoided before synthesizing a compound to avoid DILI? This came at the same time as the transporter discussion, but I presume it's an open question for transporter inhibition as well as for reactive metabolism. So Mike, go first.
Sorry, Kaushik, I was muted. Yeah, no, thanks for the question. It's a really good one. So, I point the audience to a recent publication in Journal of Medicinal Chemistry entitled Drug-Induced Liver Injury Mechanisms and Medicinal Chemistry Avoidance and Mitigation. And what the paper does just provides a really high-level overview of some of the SAR around these different endpoints. So whether it's BSEP inhibition or mitochondrial toxicity or reactive metabolism. Speaking to the BSEP part, I think the literature data to inform on SAR trends for BSEP inhibitors is immature, but it's emerging. I think the problem is there's been several publications on BSEP inhibition, but the cutoffs that they use to define inhibition have really varied widely, right? So one group uses 25, one group uses 300, and then when they publish on, okay, this is the SAR, if you're using a cutoff of 300 and you're saying, okay, well, C log P greater than 2 will get you potency, less than 100, and you'd have to worry about it, right? What do I actually do with that number? It seems as though that things like molecular weight, lipophilicity, higher heteroatom counts have some relation to BSEP inhibition, but of course these things affect other parameters in drug discovery and development, and so these parameters are often optimized to build in potency or selectivity or avoid other transporters. So I think it's a field that's immature but emerging, and right now it's difficult to really understand how to get out of this space. The paper actually recommends frequent testing of SAR in the in vitro assays to better understand how to get out of that space, in the structural series you're working at.
Thank you, Mike. Jim or Wen, do you have any thoughts on reactive metabolite alerts?
This is Jim. I think just a final note for me would be, I think we've all kind of emphasized it, the link that we're trying to make to efforts for rational drug design, right? The word prediction, I think, is very seductive when you see, the de-risking literature out there, and ultimately, this idea that one assay can't stand alone, and that we're really trying to work with all the different functions of the drug development apparatus at Merck to help define better structures, better scaffolds. Thank you.
Great, no, thank you. All right, so I have another question for you, Mike, to say that the question is from Brian around, do you run a TDI or any kind of inhibition studies with BSEP to look for effects of metabolites?
Yeah, so another interesting question. You know, it's on our list of things to do. I think it certainly is intriguing whether or not you could potentially have a TDI against a transporter, but it's an area that we haven't focused on yet, and I don't think other people have investigated it very thoroughly either. So unfortunately, can't really comment more than that on that question.
Would
But
you
it's a good
say
one.
that would you say, Mike, that having this in a metabolically competent system might cover it?
Yeah, I mean, it could if it's within, you know, in that time frame. I think you probably could catch it. I think you'd have to, you know, it would be good to see a positive control in the system if we
Of
can
course.
find one.
Yep. Yeah. A question from Shelley on whether there is a large BSEP inhibition data set that is compounds and the associated BSEP inhibition activity. Are those data available in public domain?
Yeah, another good question. I love that we're getting a lot of questions on transport. It's awesome. There's been multiple publications that look at extensive data sets. So there's been publications from Amgen, AstraZeneca. We have published. I don't think you can find all those data in one place, although there has been efforts to kind of combine it all. Like I just gave that paper a little earlier around some of the chemistry aspects of it. There's also a white paper on BSEP inhibition that we published in 2018 with references to all the paper. Unfortunately, you have to do a little bit of digging, but certainly I think there's been a lot of work done in the field in the last 5 years.
Very nice. From Barry, another question on BSEP inhibition. Is atorvastatin a BSEP inhibitor in vivo under standard dosing conditions? Or is it just an inhibitor in your in vitro study which uses high concentrations?
Yeah, thank you for that question. Yeah, so we're using it at a very high concentration to inhibit. Actually, I think the exposure clinically, the maximum exposure is like 200 nanomolar, the unbound concentration. That's in a worst case scenario where you're assuming everything is absorbed, right? So it's at FU, IN max, or the free inlet concentration. The BEI for atorvastatin, I think, is 35. So you have quite a large margin between the free inlet worst case concentration and the BEI IC50. So I don't think there's any publications that I'm aware of that tell us that atorvastatin is a BSEP inhibitor in the clinic.
Yep, agree with that. How do you assess the AUC of the media in the Patopac assay? Can you provide more information on time points, etc.? This is for the both the BSEP and the I think when you covered this in your part where you discussed the time points, but for the Patopac assay regarding BSEP, do you have a sense of AUC or are you going by concentrations? Mike, this is a question from Caroline.
Yeah, that's a good question. So I think it's been published several times from Kim Brower's lab that the change in taurocholate concentration using a 1 micromolar dose in 10 minutes in these hepatocyte systems doesn't change. So that AUC is actually a constant. So we're actually not measuring it. We're just assuming it to be constant. And I think that's supported by various publications in the literature.
Large amount of questions on transporters. Are there any drugs that inhibit NTCP and BSEP? This might account for some of the drugs that inhibit BSEP but do not promote DILI. Question from Barry.
Yeah, so another good question. There certainly are compounds that that inhibit both NTCP and BSEP, and that makes going to clinic, you know, oftentimes they try to measure increases in plasma bile salts, but they're thinking that if you inhibit BSEP, you then get these compensatory mechanisms which will pump these bile acids out into the plasma. The issue with that is that, yes, you can get increases in plasma bile salts from inhibition of BSEP, but you can also inhibit NTCP. I think what our data shows is that, you know, I showed you several compounds that inhibit in vitro biliary clearance, and that's telling you that the uptake is inhibited. So what we're finding as we start to think about translation is that many compounds inhibit both NTCP and BSEP, and maybe that's not really so surprising as they're probably evolutionarily linked.
Yeah. I'm going to send you a different question on so the person who asked the question is no longer online, so I'll send you the question by email for response. Frederick, from Frederick, this question is around covalent binding as a strategy for pharmacology. So I'm going to hand it over to Wen and Jim for this response. Covalent drugs go from fringe field to fashionable endeavor. This is a recent CNE an article. Designing molecules that make bonds with the biological targets is now in vogue. Do you guys have a thought on how covalent binding as a pharmacology strategy might hinder or might change your thinking around DILI through protein covalent binding? Wen?
That's a really good question. I think I think Jim can add more. I think that's a struggle we have, especially, you would think your pharmacological target covalent binding is very selective, but I think that's a very tall bar. Obviously, for our assay that you say we will have trouble actually to separate those. I think there's some work we have done. I think Jim in his paper had talked about, if you look at the pathways, we think adaptive pathways that response to chemical insult, like the nerve 1, nerve 2, perhaps there's something there we can look into further, perhaps help us to dissect out those details, if you will. Jim, I'll let you speak.
Yeah, it's a really great question, and one that we struggled with. I personally make the distinction that I think Gwen alluded to. There's a difference between promiscuous covalent binding and targeted covalent binding. And so I think the more partners you dance with, so to speak, the greater the risk probably, and the greater likelihood that the mechanisms, the defense mechanisms that you trigger in the cell, the threat that the cell perceives, the greater that will be. So yeah, I think that the better you target your covalent binders, the better. The thing I'll add is that, we've had some cases where we've actually done some SAR around the accompanying structure, right? So these things typically have a reactive warhead. So, what do you see when you take that warhead off? Are there additional risks in the molecule, hotspots in the molecule that may additionally contribute to bioactivation risk? I think that's a question that we try to ask when we get these sorts of cases.
No, that's an excellent answer. So essentially what you're saying, Jim, is because of pharmacology, you can't really avoid the protein covalent binding coming from the target part of the molecule, but you would take caution around other parts of the molecule that might get bioactivated.
Absolutely.
Yep. Thank you. This is actually an interesting question from Frederick on how MIST relates to DILI, MIST guidance as in the metabolites and safety testing. Any thoughts, Wen or Jim?
I, speaking as a non-drug metabolism person, I think that's a really good question to think about how we sort of think about the risk, because, like we said, certain things, for example, unstable acyl-glucarate, so that clearly is something that, if you look at the guidelines, that's something you do, but I think we still try to understand how that's related to DILI for specific compounds and cases. I'm not sure if that's helpful.
Yeah, I know what you're saying. I think MIST, as it's defined in a regulatory perspective, is about a certain cutoff from the for metabolites circulating in blood, right? That's sort of the guidance for MIST. But the thing to remember also is that the site of DILI is obviously reactive metabolism in the liver, And these are metabolites that you do not see circulating. These are reactive and they bind to proteins. So apart from the example you just gave when in acylglucuronides, which may have maybe reactive acylglucuronides and may move around in the body to target organs, although not DILI, it could be something manifested in the skin, for example, right? So that would be looked into. Obviously, so circulating metabolites that are reactive as acylglucuronide would certainly play a role in de-risking strategies when you're trying to put a compound in the clinic.
Yeah, thank you, Kaushik, for articulating that very well.
No,
Thank you.
Yeah, no, thank you. So sure, this is a question from Sean from one of my colleagues at Janssen. The in vitro hepatopac LRA has a 9-day treatment. Have you tried to identify marker genes at earlier time points, say day 1 or 2, for obvious reasons that you can cut down an early screening assay to a shorter turnaround time for the chemist?
Yeah, as I have to say, we have done limited. I think to our surprise, perhaps, you would actually get a lot of response if you use a shorter assay, but for some reason, we would start to see a lot of drug we consider safe start to have a response, which we do not fully understand. Perhaps, you know, the maturity of the platform, maybe one, or, you know, the cells, even in vitro, maybe they do mount adaptive response and readjust. So that's something we do not fully understand. Clearly, it's a really good question. I think it's worth looking into.
Great, thank you. I think that's the end of our questions, David, if you want to take it away.
Okay. Hi, this is actually Russell, and thank you, Kaushik, and then James, Mike, and Wen for your time and insights regarding efforts to mitigate drug-induced liver injury today. We'll be taking a break next week for the Thanksgiving holiday here in the US, but we do invite you to return for the first week of December. On Tuesday, December 1st, join Jim Skinner of Terrangina Incorporated and the ACS division of small chemical businesses as he discusses the different types of investors, their investing criteria, and the impact that their investment will have on the ownership and management of your company. Then on Wednesday, December 2nd, join the editor-in-chief of the National Journal's Hotline for a recap of the 2020 election. You will hear how the coalitions for Republicans and Democrats are far from being clearly defined ahead of 2022 and 2024, and what that could mean for the chemical industry. The American Association of Pharmaceutical Scientists is a professional scientific organization of approximately 7,000 individual members and over 10,000 actively participating stakeholders employed in academia, industry, government, and other pharmaceutical science-related research institutes worldwide. Find out more at aaps.org. And if you are a medicinal chemist or interested in the field, we invite you to join ACS Medi. One of the many benefits is the annual reports in medicinal chemistry, an outstanding roughly 600-page volume, providing timely and critical reviews of important topics in medchem. We'd love to hear what you thought about our program today. You can reach out to us through the ACS social media channels on Facebook, Twitter, and LinkedIn, or by email at acswebinars@acs.org. A short survey will pop up directly after the webinar that will take only a minute to complete. Also, remember that an invitation email to view the unedited recording from today's presentation will be sent to all registrants for a 24-hour period, after which it will be removed, edited, and become an ACS member benefit. As a reminder, ACS Webinars does not endorse any products or services. The views expressed in this presentation are those of the presenter and do not necessarily reflect the views or policies of the American Chemical Society. And that wraps us up for our program for today. On behalf of all of us at ACS, thank you for joining us, and we hope to see you again soon. Until then, stay safe.
Thank you.